Dear chairs, dear guests, dear students, thank you first of all for the chairs for the nice introduction. And I would like to show basically the activities of my research laboratory in the Biological Research Center. And we are very much centered on biological barriers. So the topic of my talk is why biological barriers are important. We have many barrier systems in the body. These can be outer barriers like the skin, like the cornea at the front of the eyes, or like the epithelium lining, either the respiratory system or the gastrointestinal system. You can see that the red ones are showing the kind of barrier systems we are working with. We also work with some inner barriers which are separating not the outside environment and our bodies, but the blood from certain inner organs or systems. And from these ones, we study the so-called blood-brain barrier. In general, barriers are important because they provide protection against harmful effects. Whatever these effects, we can see that one of them are the pathogenic microorganisms that we found in the outside environment, and these barriers provide physical barrier. You can see here that these tight junctions are the main components of these physical barriers that do not let molecules, cells, enter between the cells. Here you can see one microbe, it's a bacterium, and this is the virus of AIDS, the so-called HIV virus. Another aspect of this protection is the so-called chemoprotection. So not only from cells or microorganisms, but we are protected from toxic molecules. So in the previous talk, you could hear from Dr. Attila Hunyadi that there are a lot of molecules, some of them are toxic, for example, what we use against cancer cells, and the cells protect themselves against these substances, and what you see here, these are the efflux pumps. Efflux pumps are a group of proteins. These are the ones which are catching such kind of usually amphiphilic or lipophilic molecules, and basically they are pumping it out. One big example of this class of pumps is the so-called P-glycoprotein, which is very, very interesting from biomedical point of view. In general, the two main reasons why barriers are important is because they are limiting drug delivery to the body and within different organs in our body, like they are limiting the entry of water-soluble molecules, especially biomolecules, biotherapeutics, and because of the previously mentioned chemoprotection, they are limiting the entry of the so-called efflux pump ligands, which can be cancer drugs, antibiotics, and many other class of therapeutic molecules. This is the reason why it is difficult, first of all, to give, for example, biomolecules orally, like a tablet, and nobody likes injections, which is normal way of giving these medicines. And second, it is difficult to treat the neurological diseases because the blood-brain barrier protects the brain tissue. So it's very difficult even from the brain to get through this barrier. The second major reason why we study barriers in general is because diseases have an effect on these barriers. I just give you an example, the example of the blood-brain barrier. So even systemic diseases, that means like alcohol disease, diabetes, or infectious diseases, like AIDS, which is affecting the brain, so-called neuroAIDS, they do have an effect on the function of this barrier. And also we can tell that in many neurological diseases, the function of the blood-brain barrier is seriously impaired. This includes Alzheimer's disease that many of you have heard about, also epilepsy, which is a disease which is affecting about 1% of the human population. And also diseases like brain tumors or stroke or traumatic nervous system injury. Now, the question is how we are studying these diseases. In my laboratory, the model systems include culture models. Here I give you an example about the different epithelial culture models. So we have usually different cell lines of epithelial cells. These are in liquid, and we are seeding them on porous membrane containing inserts. These are plastic cylinders with a porous membrane at the bottom. The cells we are adding to the liquid, they just sink, attach to the surface of the membrane, and form a confluent layer. The cells are attaching to each other without any gap. When we do this, our model is ready, and we can control this kind of barrier formation by measuring the electrical resistance across this layer. This is a very good index because it means that the layer is so tight that even tight for ions. And this is the model we are using also for checking the permeability of the molecules, which is very easy. We have two compartments. We have a compartment above the cells, we have one below the cells, and basically the cells on top of the membrane separate completely these two compartments. So any molecule from the top compartment to the bottom is crossing the cells. And we can calculate basically the permeability of the different compounds. And actually we do a lot of such studies, either for testing new molecules, like in the case of the molecules that Attila Hunyadi was mentioning before, or we do the same test, we are testing molecules for companies too. We not only work on epithelial barriers, but we also work on blood-brain barrier, which is brain endothelial cell layers surrounded by pericytes and astrocytes. This is a very complex 3-cell type model that we use to mimic anatomically what we have at the level of the tiniest blood vessels in our brain. And this is the model system that we have described together with our Japanese partners, and we are using these model systems also for testing molecules and also for testing, for example, protective effect of different kind of molecules. The next step in our evolution of culture models was the development of a microfluidic and electronic barrier chip device. And this is very similar that it is also a two-compartment, a top, which is the light blue, and the bottom, which is the pink on the figure compartment model. And this is also based on the same principle. So we have a porous cell culture membrane, and we can basically culture the cells on this model. The big novelty of this device is that we are introducing flow, fluid flow, because in our model system previously we used a static model, so the medium was basically static, but we know that in our vessels, blood vessels, the blood is flowing. So we need the flow to stimulate the cells with the shear. And this is what we do. So we have a setup in which we are just feeding the cells, and then we have a dynamic setup when basically the medium through a peristaltic pump and the reservoir is basically in the upper compartment is circulated. And what we are using these models, these culture models, so basically our major research areas are the following. We are studying drug delivery across different barriers. We know that the junctions between the cells stop the passive permeability, so we try to open these junctions between the cells. And the second major way of achieving higher drug permeability is targeting this kind of drugs through nanoparticles. And not just self-assembly nanoparticles, but we try to make vesicular nanoparticles, as I will show later. And also we are very much interested that if the blood-brain barrier is damaged in different kinds of diseases, how this damage is happening and how we can protect against this damage. So the modification of the BBB tight junctions I was mentioning is the following. For example, we were testing short-chain alkylglycerols, and when we were adding them, we could see that the resistance dropped, but when we removed these alkylglycerols, the resistance returned within minutes. Then we studied different specific peptides, which were targeted against different protein motifs of the junctions. And then we could also see that peptide treatment resulted in a drop in resistance, and by removing the peptide, the layer resistance very nicely returned. An example of our targeted nanoparticle approach is shown here, when we were using red fluorescent nanoparticles, which were either targeted with biotin, these are the little yellow dots here, or glutathione, these are the red Pacmans. And what we could see here, that the uptake of brain endothelial cells was increased with biotin and glutathione as compared to the non-targeted, the first nanoparticles. And not just the cells took up more nanoparticles, but more particles crossed these layers from the top compartment to the bottom. That means that these were going across the cell system. So we were really very confident that both biotin and both glutathione can be used as targeting molecules. In our next series of experiments, we were using vesicular particles. We had a big biomolecule, albumin, as a cargo. And we again had a non-targeted particle. And then we had alanine, glucopyranose, a glucose analog, and glutathione, the tripeptide glutathione. By the way, it's an antioxidant. But the SH group is not playing any role. So the SH group is used for fixing it to the PEG. And double targeting ligands like alanine glucopyranose, alanine glutathione, and glucopyranose glutathione. And you can see again the model I was telling you before. So we could see if we put the nanoparticles in the top compartment and we checked what is crossing the cell layers, and we see that compared to the albumin molecule, the single ligand and the double ligand targeting particles cross the layers much better. Among the protection of the blood-brain barrier, we were using some clinically used medicines, like the antioxidant edaravone that is used in Japan as a post-stroke therapy, and some sulfated polysaccharide heparin analogs, the pentosane, which is used also, mainly used as a cream for different venous problems in the leg. And then we also use some phosphodiesterase inhibitors, and all of them proved to be really cytoprotective. But we not only tested medicines, but we also tested some food components that everybody except underage minor can have, because here is a red wine, but resveratrol, which is a polyphenol from grapes, you can get it through eating grapes. So not only alcohol is rich in resveratrol. And we were also very happy to show that some of the fish oil is also very, very protective for the blood-brain barrier. We have many international cooperation partners, not just in Japan, but also in Europe and also in the USA and Mexico. And we are also involved in a very complex project, including partners from Taiwan, Luxembourg, Israel, Austria, and Switzerland, in which we would like to study nanoparticles in a chip device using midbrain organoids derived from stem cells. And the goal is to develop very specific nanoparticles which can be used in therapy in the future. So these are the current members of my team. And we are very, very proud of Gergő Porkolab, who is a senior year student, and who started his research work as a summer student practice in our laboratory. And he got, after really hard work, a lot of very nice prizes. And I just would like to emphasize that in this picture he received the Kufla Prize this October, and we are really happy for the foundation and also for the possibility that we can work together with Gergő. With that, thank you very much for your attention.